Welcome to the video for chapter 9.5. We're going to talk all about microbial taxonomy, or at least an introduction to the topic here in chapter 9.5. This is going to be important because as we move into chapter 10 is talking about the different phyla of bacteria and some of their characteristics. So we need to kind of learn what that means. Before we start the material for 9.5, let's do a review question real quick. Review question number 1 says, which form of horizontal gene transfer requires cell-to-cell contact? Is it A, transformation, B, conjugation, C, transduction, D, conjugation and transduction, or E, all of the above? So take a second, pause the video, go and flip through your notes for chapters 9.1 and 9.3 and see if you can't find the answer. Okay, the answer for this is B, conjugation. Conjugation is the only one that requires cell-to-cell contact. Okay, so transformation is literally the bacteria is going to pick up DNA from its environment. Conjugation is going to use the conjugation pilus to pull a donor or a recipient cell closer to the donor cell. Transduction is going to use those bacteriophages and use that virus-mediated transfer of DNA. So the only answer for this would be B. Okay, so 9.5 is all about microbial taxonomy, or at least the introduction to microbial taxonomy. You probably could take a whole course about microbial taxonomy, to be honest. Okay, so some of the section objectives we have for this lecture is we'll be able to describe how we define distinct species of bacteria and explain why the definition of species is problematic. We'll be able to explain how microbe is identified and describe the relative advantages and limitations of a dichotomous key as compared with a probabilistic indicator. Okay, so let's take a moment and let's talk about how we classify microbial species. Now, I don't know if you've noticed this about scientists. We love to name things. We love to group things together with other things that look and behave just like that one thing. Okay, in fact, for living things in biology, we have a whole science dedicated to grouping things together and putting them all into their own little boxes. It's a science called taxonomy. Okay, taxonomy is the description of distinct life forms and organizing them into different categories with shared traits. Taxonomy is one of those things that we've been doing for a very, very, very long time. It consists of 3 parts. We have classification, phylogeny, and nomenclature. Okay, so classification, this is the root of the whole science. This is literally putting life into different groups by shared traits and shared behaviors. Okay, those groups we call taxa. Okay, so this is where you look at 2 organisms and you try to see how related they are to each other. Or you discover this new species, what do we know that looks like it? Okay, so obviously things like tigers and lions, they look very similar to each other, right? They're both big cats. So obviously those got put into the same box. Now, maybe things like plants and fungi, right? They don't move, they look, or they're organic, they look like something you could eat. Maybe they're in the same box too. This is what classification is literally looking for shared traits and classifying things by those traits. Now, taxonomy in the last 30 or 40 years has added a new branch called phylogeny. So phylogeny is the evolutionary history of organisms and how related to each other they are. Now, this is one of those sciences that has gotten really popular in the advent of things like PCR and DNA sequencing. We use phylogeny all the time now because we literally can take an organism and we can sequence this genome and we can look at every other genome we've sequenced and see how related to each other they are. In fact, a lot of our classifications changed a lot once phylogeny and DNA sequencing came on the scene. In fact, I can remember in my first taxonomy class in college, granted this is 2005, 2006, so not too long ago, we were learning how to classify things, and back at that point, we were still classifying fungi and plants together, okay? Because based on their looks and some of their behaviors, we thought that they were going to be grouped together. They'd be part of the same taxa. But once we started sequencing the DNA of different organisms, we found out that plants and fungi aren't really related to each other. even though they look very similar to each other. In fact, animals and fungi are more closely related to each other than plants and fungi are. So me and you have more in common with a fungus than plants do. Okay, so phylogeny is looking at that fossil record, the evolutionary history, and looking at how things could be related to each other. This is going to go more into that tree of life, that phylogenetic tree that we've seen in several science classes, where we have the common ancestor as the trunk of the tree, and everything starts to branch off from that common ancestor. This is what phylogeny is. Okay, so how we're related, what branch are we on, and so forth. Okay, and the thing is, with taxonomy, if we are going to spend time putting things into boxes and classifying things, we need to have a common naming system, right? We have to make sure the names make sense. Okay, so this is where the nomenclature part of it comes in. Okay, so we have a standardized system on how we name living things. Okay, in fact, the scientific name for things is always the same. It's what we call the genus and species of their taxa rankings. Okay, and we'll talk a little bit more about this on the next slide when we can see an example. Okay, so this is really easy to do with living things like animals, but what about microbes? Okay, so microbes, if we look at some of them underneath the microscope or on plates, they look very similar to each other. In fact, if I could think of about 5 bacteria off the top of my head that we could look at it, and we would think they were all the same bacteria. Okay, so how do we classify these? Okay, for a lot of animals and other living organisms, we can classify them based on the fact if they can reproduce with one another. So if two organisms can reproduce, they're probably in the same species. So like horses and donkeys, they're in the same family, they're in the same genus, they're in the same pretty related species. They can reproduce with each other, right? A donkey and a horse can make a mule. Okay, lions and tigers, they can have ligers. Okay, we really can't tell that with bacteria because bacteria go through binary fission. So what way or what ways can we use to classify them when they all look similar to each other? Okay, so if it's so hard to classify microbes, how do we do it? Right? We know things are grouped together, right? We have gram positives, we have gram negatives. How else do we group things together? Well, when we're classifying microbial species, we use 2 factors. We use phylogeny and we use ecology. So phylogeny is literally how related are they by their DNA, by their genomes. Okay, it's really easy to take a bacteria, a microbe, and sequence its DNA and get its genome. Okay, then we can then put it into a database and see what else it's related to. Okay, so that's going to be able to help us figure out its taxonomy. Okay, so like here we have an example of the taxonomy of a certain species of bacteria. Okay, so all life is divided up into 3 domains, right? Bacteria, archaea, or eukarya. Obviously, a lot of the ones that we are going to be talking about are bacteria. Okay, with animals and eukarya, we usually have a kingdom after domain. So for bacteria, after domain, it's phyla. Okay, so these are going to be a less broad category than the domain. Okay, we get more specific as we move down the taxa. Okay, then we're going to have the class, the subclass, the order, and the family. And then finally, the genus and species. Now, the genus and species are really important. This goes back to that nomenclature part of taxonomy on the previous slide. We use these 2 to identify different organisms. This is our common method for naming something. Okay, so like Staphylococcus aureus, Staphylococcus is its genus, its species is aureus. Okay, Staphylococcus epidermidis, so Staphylococcus aureus and Staphylococcus epidermidis are both from the same genus, but they're different species in that same genus. Okay, so genus and species are that common nomenclature, the common way that we name things in science. Now, I will say we really aren't going to talk about class, subclass, order, family in this class. We're going to stick mostly with domain, phyla, genus, and species. In fact, the next chapter, in chapter 10, is all about microbial diversity. We're going to talk all about phyla in that chapter. Okay, so that's phylogeny. Let's talk about ecology. Ecology is any shared traits and shared ecological niches that these microbes might have. Are they aerobic? Are they anaerobic? Are they fermenters? Do they ferment lactose? Do they not ferment lactose? So what do they share environmentally that we can classify them? How can we group them that way? Okay, so this will be on the exam for sure, 100%. Okay, the question will ask you something like, how do scientists classify bacteria? And your answer is going to be by using phylogeny and ecology. Okay, well, even then, with these two factors, identifying a microbe is hard. Okay, we can't see bacteria with our naked eyes. We have to use a microscope. Okay, the best way to figure out and identify a microbe is to use DNA sequencing to look at its genome. But the thing is, not everywhere has access to this DNA technology. So how do we do that without that? Okay, I will say in the last 10-15 years, most medical practices are starting to get the technology and using more and more DNA-based methods in their practice. But not everybody can afford to get a DNA sequencer. They don't have staff that can run a DNA sequencer. So what do we do if we don't have access to that? Well, we're going to use two things to make the identification of a microbe. Okay, we're going to use phylogeny, and we're going to look for phenotypic traits, so things we can observe. Now with bacteria, because we have to use a microscope, microscopes are going to come into this, or it's all part of identification. But what else can we use as a phenotypic trait? Well, we're going to look at things like shape, elevation. So a lot of these things that we did in lab 5 with those, or was it lab 4? In lab 4. Okay, when you went through and you used all those terms to describe those colonies on your plate. But an even better way is we're going to use something called a dichotomous key. Okay, so a dichotomous key is going to be a flowchart that consists of either or yes, no questions that we can answer, and they're going to help us identify the microbe based on things that we can notice. Okay, so one of the things, a lot of them start with gram-positive or gram-negative. Okay, where did we find that? Okay, so this dichotomous key is specifically towards wastewater. Okay, so we did a gram stain, we found out that our bacteria is gram-negative. Okay, so we're going to use this dichotomous key. The thing about dichotomous keys is they're really great, but they're not something that somebody off the street can come in and use. It takes a lot of training in order to read a dichotomous key. It's going to use a lot of microscopy in order to do so. Okay, so let's kind of walk through this. So we have it identified as gram-negative. Okay, we have something that's less than 1 micrometer wide, and we have something 1 to 3 micrometers wide. So if we answer yes to this side and follow the branch here, we really don't know what it is. It's not big enough to identify. But if we have a width between 1 and 3 micrometers, well, that moves us to the next question. Is it filament straight to smoothly curved, or filament coiled? I really don't know what these mean. I've used some dichotomous keys in medical microbiology. This one I'm not familiar with. Okay, so if it's filament coiled, it's going to be this bacteria here. Nostocodia limicola 2. Okay, well, if it's filament straight to smoothly curved, well, that gives us some more options. Okay, does it have a sheath or does it have no sheath? Well, if it has a sheath, is it follically branching or no branching? Okay, it's going to give us either one of these two. Okay, well, maybe it has no sheath. Well, that gives us a lot more options. Now, I want to pull your eyes down here to these ones with numbers. Okay, so these are bacteria species that have not yet been identified, so we don't know what they are. Okay, but this is how we would use a dichotomous key. We would just answer yes or no as we go through this. So you can kind of see why it would take a lot of training in order to use this dichotomous key, because you need to know what these mean. What's filament coiled? What's filament straight or smoothly coiled? What's a sheath? Okay, what's this false and no branching? I don't know what this one is either. Okay, well, this one's a little bit easier. Motile, okay, it's moving. Okay, does it use sulfur granules or no sulfur granules? Okay. Is it rectangular? Is it square? Is it barrel? Who knows? I don't know what those mean. But this would be one way to identify a microbe. Okay, there are some disadvantages to this. This takes a lot of time. It takes a lot of knowledge and a lot of training in order to use one of these dichotomous keys. Now, a better way would be to use something like a probabilistic indicator. Now, what this is this is a metabolic test. Okay, so we are going to put our sample into these 96 wells, and we're going to run these 96 chemical tests all at the same time. It's all based on metabolism. So when we run this, each microbe is going to have a very set result. It's going to be kind of like a fingerprint. Okay, so we can use this test to identify microbes with more accuracy. Okay, and one of the best things about this is anybody can set this up, and most of the time the reading of the probabilistic indicator is done by a computer. Okay, in fact, it will look at the results. Like, let me find one. Okay, so this is whether it survives in an acidic pH or not. Does it produce lactic acid? Okay, is it gram positive or gram negative? Okay, so all these different things are things that we can use. The computer is going to take these results and say, oh, it's a 93% 93% chance that this is Stenotrophomonas multifila. Okay, so it's going to give us the probability that it's that certain species. It's not always 100%, but this is a way easier way in order to identify things if we don't have DNA technology right there. Now, this is way better than dichotomous keys. You don't need as much training. Anybody can do it. The computer reads it for us and compares it to a database. So there's not any real disadvantages to using a probabilistic indicator. It is much, much quicker than using a dichotomous key as well. Okay, so that is 9.5. We've talked about taxonomy. We've talked about how we can identify microbes based on both DNA tests and phenotypic traits. Okay, this is the end of chapter 9, and we'll see you for chapter 10.